Deep, let me know when we are live. Okay, then starting. Great. Hello, everyone. Greetings from Biologically Speaking, and a warm welcome to the second day of Scimplify 2021, Simplifying Science for All. Very good morning, good afternoon, good evening to all, wherever from you are listening to. I'm Sundaram, a graduate student, currently at Csir Institute of Genomics and Integrative Biology, New Delhi. I'm a co-founder of Biologically Speaking, and I'll be moderating today's session. Biologically Speaking is an academic interest group currently sponsored by the American Society for Cell Biology, Compass Outreach Grant, and designed to showcase the global frontiers in biological research. For last 9 months or more, we have been conducting biweekly seminars, and we have joined by eminent scientists across the world, and nonetheless have garnered overwhelming support and love from you all. And thanks again for making our humble effort a huge success. For more information, do visit our website, www.biologicallyspeaking.com. On the auspicious occasion of World Science Day, which has been celebrated across the world to showcase the significant role of science in society by engaging public dialogue on the current scientific issues, and also it underscores the importance and relevance of science in our daily lives. Simplifying Science for All is a humble effort from Biologically Speaking to spread the world of science to masses by simplifying the apparently complex scientific world, either by actively engaging them in science or by exposing them to the highest level of science by the eminence in the field, with the vision of developing scientific temper in the society and also instill the curiosity of natural world in the mind of young generations. In the current session, All Roads Lead to Biology, we will try to capture the interdisciplinary nature of biological sciences, which is very explicit since its inception. For a moment, if you consider the discovery of the structure of DNA, essentially the code of life, one of its discoverer, Francis Crick, started as a physicist. And also our very own, like Dr. Hargobind Khurana, Dr. Venki Ramakrishnan, all we know, both won a Nobel Prize in their respective research on biological problems. And they started their career as a chemist and physics, respectively. And there is no dearth of such examples. So today in this session, we are joined by 4 distinguished speakers who have started their journey in a different domain, yet finally delved into biological research to solve the biological problems and contributed significantly. Today, they will be sharing their journey and experiences with us, followed by a panel discussion. So before we start, a couple of housekeeping things. If you have any questions for the speaker, please enter in the Zoom chat box. Those who are watching on the YouTube, please type your questions in the comment section. We'll try to moderate these as many as possible after the talk. So without further ado, let's start today's session. All roads lead to biology. Our first speaker in this session is Dr. Anurag Agarwal. He's a clinician scientist who is currently director of Csir Institute of Genomics and Integrative Biology, a premier national laboratory in India. He's a pulmonologist by training, interested in understanding biological and clinical aspects of respiratory diseases. His outstanding work has received many accolades, including Shanti Swarup Bhatnagar Prize in 2014, the highest Indian science award for his contributions. Dr. Agarwal is deeply engaged with the science society interface and biomedical policy development. His current focus is use of information technology and big data analytics for effective application of community health efforts and development of pattern-based diagnostics. So with this, stage is yours, Dr. Agarwal, over to you.
Thank you, Sundaram. a pleasure to be here with all my distinguished colleagues and young colleagues perhaps in the audience. Let me share my slides and we will go from there. Give me 1 second and here we go. So when Sundaram, you asked me to speak on this topic today, it was quite interesting because in some sense, if I look at my journey, I have never really shifted. I mean, medicine and biomedicine and biology and medicine are quite similar. So unlike some of my colleagues who really made a big lateral shift, to me, it's been a question of being in the same field with a low fence that you kind of just hop over. But I do recognize that the field of medicine and the field of biology are often separated and sometimes for no really good reason. So this is the problem the way I see it. There is conventional medicine that we think of when we think of medicine, and there is biomedicine. And really speaking, biomedicine is what I always felt I was doing, which is why going to biology was a very small jump. But what are the basic differences I see that perhaps I had to emphasize more in my career and convince other people? So, there are always heroes and there are sidekicks. And I think the picture I have on this slide will be very, very familiar to all of you, with Robin the Boy Wonder, a grown man but without long pants, standing alongside Batman. And conventional medicine does not see itself as derived from biology, derived from basic sciences, but rather sees it as sometimes as a sidekick. It also needs to be done. The only difference between biomedicine and medicine fundamentally is about research as being something essential, the source of medicine, and not a sidekick. So the minute you accept that, you move into the idea of a culture of questioning, experimenting, accepting, trying again, accepting that you'll be wrong. You can do none of this unless you have knowledge of and access to the frontiers of biology. I would even argue that simply knowledge of without access to is good enough to build the mental mindset that is required to at least be accepting towards the limitations. For example, I often tell people who join my lab that half of what you know in medicine is false. And the trouble is you don't know what half. So for example, when I joined medical school, if I had given beta blockers, something that slows down the heart and reduces its contraction, to a patient with heart failure, they would have failed me. By the time I finished my residency in internal medicine, MD medicine, if I had not given beta blockers to somebody with heart failure, they would have failed me. So, these are polar opposites, from absolutely contraindicated, or at least relatively contraindicated, to main line of therapy was a big change. Which then basically requires that you must have better connection to research institutes, and you must have career tracks for true physicians who become scientists as opposed to academic clinicians. So, when people talk about how to make this shift, they talk about this very nice circle. You go from basic science to clinical settings, you can call this bench to bedside, and you go back from bedside to bench. You could have a clinical question, suboptimal outcomes. You could have a biological question, curiosity. But in real life, it looks like this. I mean, that's really what it feels like when you try to do these things. When you begin addressing simpler questions like I did early in my career, it was actually quite simple. And as a personal perspective regarding what possibly makes me say what I say, my interest was primarily in computer science engineering. I joined AIIMS Delhi for MBBS along with many other confused people. The selection system those days was such that the AIIMS entrance exam had a lower weightage for biology than for physics and chemistry. Pursued higher training in medicine, pulmonary disease, critical care, spent some time in ICUs treating people, shifted to experimental research, and most recently shifted to data science, public health, which brings me back towards my original interest, that was computer science. But to show how everything comes together, let me highlight this one experience that, to my mind, changed the way I think about things. Now, if you want to measure the product of the resistance of the lung and the volume of the lung, and you can imagine this is important, right? People with severe lung disease have high resistance like COPD. They have hyperinflation of their chest. Measuring these things are important, but only their product could be measured easily, not them individually. To separate them, you put people inside what we used to call a body box. You close a shutter so the person can't actually change their lung volume, and you make them pant. And that's like it's pretty hard for sick people to do it. And being an asthmatic myself, I had to do it in my father's lab. And it was tough during the time when it needed to be measured. So the time you need to measure it, it's tough to do. When you feel fine and nobody needs to measure it, it's easy to do. That's the beauty of many things, including blood pressure. So what we thought on a walk, my father and I, towards the end of my medical school, is rather than doing all those complicated things, if you simply do algebra, you insert a new resistance that's called R additional, INS is instrument. You make two measurements, then you do simple algebra, you can calculate all the separate components. And to do that, now you do a little bit of engineering. So what you do is you put a shutter here, that's a resistance, both are known. First, the person breathes this way. You close the shutter, he breathes like this. And that shift instantaneously will lead to a change in SRAW. And by measuring it twice, you could do it. It was my first paper in life. And I must say that this was the only paper for many years that ever went into one of the higher journals in the area with not even a single word changed, except for the reviewer commenting, why didn't anybody else think of that before? You can see there was nothing very complicated. There's a little bit of engineering design, implementation, a little bit of basic mathematics, but mostly it is about looking at things in a fresh way, and it tells you there's always much scope to improve upon what exists. Large blind spots exist between eyes of different disciplines. In this case, perhaps even within medicine, physiology and lung disease. And finding those blind spots is an excellent way to go about doing research or building research careers, which might be the bigger interest for some people. And what I realized further during the time that I went to US was, even though that particular question came, I was bad at asking questions. I was trained to be much better at answering questions than asking questions. And I would rely upon bookish knowledge to answer questions. And when a good speaker came, who's talking about the frontiers, I was rarely the person in the audience who would be asking that challenging question that would point out inconsistencies in their data. So what I learned mostly in America, but I think it can very much be learned in India, different mindset required sometimes, is to ask bigger, better questions and learning to be very, very humble about book knowledge. Research is ultimately where it starts. It may enter the books, but even the fundamental paradigms can change. And we have seen that so often in biology. So what are the advantages and disadvantages? As I shifted more and more towards research and interacted with people who did not come from my background or came from a classical research backgrounds in biology, I realized that as a physician, I had a far more solid grounding in human biology, more specifically physiology, pathology, possibly even pharmacology, which is a huge advantage in disease-centric research. And of course, there's experience. But you should not trust experience too much with the N of 1 yourself. The disadvantage that I saw, and there is clearly a disadvantage. You are more into bookish knowledge. You are more into following protocols without thinking that much. You lack wider exposure, and you lack the training in fundamental biology and mechanistic research, which can be a disadvantage. You have some advantages towards, say, epidemiological research or population research, but mechanistic research is not something that comes naturally. In my case, I trained in the US. I was allowed to do both things side by side. I joined a molecular cell biology lab, and those kinds of exposures allowed me to overcome this thing. The minute I joined the lab had a journal club, and I was assigned a very journal that I thought, how big can it be? They gave me the journal Cell. So every few weeks, we would have a journal club, and I had to update the lab on anything interesting published in the area of cell. So let's talk about how I think the future will look. I was able to get the relevant training. I was able to get exposure to mechanistic biological research. When I came back to India, I joined the Institute of Genomics and Integrative Biology. I looked at joining medical institutions, and I realized that if I really wanted to do high-end research in the Indian system, a medical school was possibly not the right place to be, especially if you want to work at the frontiers. And what are these frontiers? And can you really see them? These are the frontiers of knowledge itself. And the trouble with these frontiers is that when you try to think of the future, you can only really look at the past. And I think this quotation from Paul Valery that I have borrowed from one of my teachers and mentors, Professor Srinath Reddy, is the most apt one. You can only see the landscape of the past. You're like a man rowing a boat floating on the tide. And what tomorrow looks like, you don't really know. But the one thing that other quotation I like relevant to this is from William Gibson, a science fiction writer. He does point out that even if you can only look at the present and the past, you may see the future because it's already here. It is just not evenly distributed. I'm giving you two examples. Jan LeCun's paper 1990 on handwritten character recognition using neural network architectures. So as far back as 1990, if somebody had the ability to look ahead, they could have seen that these kind of approaches may become big. Nucleic acid vaccines were topic of reviews as far back as 1995. So it requires not just waiting for the future to occur, it requires looking around you to see where the frontiers are being established. And to my mind, that is the beauty of science. To simplify things, you must always be looking at what will drive the future of whatever field you are in and bring it back to see how that particular future could change what you are doing. If you think of optogenetics, again, a very nice example of the same. So if I were to identify, 6 areas, look forward, what would they be? Some of it is taken from this very nice article in Nature Medicine in 2019. Some of it is my own thoughts on the matter. But big biomedicine, which of course, as Director IGIB, we talk about, is going to be very important. Same will be the read, edit, write genome technologies. P6 medicine, very, very important. And, of late, we are realizing that public health is one of the Ps. So it's no longer P4 or P5. It is participatory and public health are the two new Ps. Otherwise, people talk of P4 medicine. Regenerative medicine is getting very, very interesting. Immunotherapies you've seen in cancer, you're starting to see becoming commonplace with COVID, the synthetic antibodies, all that. But perhaps the biggest amongst the future, the brain-computer interface. I'm not going to take too much more time except to point out a few interesting things. So far, when we think of gene therapy, we think of gene therapy as a disease gene. You know the mutation, you correct the mutation. We think of mutations as bad things. How can it be? Some mutations must be making things better. Some must be making it worse. The ones that make things better get selected for during evolution. At some point, they don't look like mutations anymore. But if you look around the world today, there must be many good mutations as well. So of course, the people who got the Nobel Prize in 2020 for genome editing, Charpentier and Doudna. But what is less often known is a work by Helen, which is about mutating normal genes as therapy. So PCSK9 mutations were discovered as a natural, extreme lipid lowering variation. And you can target this natural gene by CRISPR. You can target it by blocking the protein, very successful. How many more such examples will be there? When we start searching for good variations, we start considering introducing such variations. And I'm telling you with all due, care that this will require a huge amount of work and caution. brain-computer interfaces. Already we are thinking of deep brain stimulation for epilepsy management. I don't know whether you have seen some of these papers, some beautiful work in Nature. Speech synthesis from thinking. If you're thinking of what you will speak, the computer can speak for you. And of course, the potentially dark side, the Skinner box, the famous patient B19. If you must look it up if you haven't read about this in the past, how you can change behavior of people. These will be true frontiers. I mean, would it be ethical to change a patient's behavior to get them to quit smoking or drinking? If that involves some degree of brain control or mind control or changing what gives them pleasure, where would you draw the line? Where would you think about the ethics of many of these areas, including genome editing in the previous slide? Which tells you one more thing. One of the biggest new important things coming is the dissociation of science, especially cutting-edge science, with humanities has to go. The future will belong to the places that bring all sciences, including humanities, thought of as a science, together. And that, to my mind, is the last message I would leave you with today, except for this last one, I guess. We're all frogs in a well. It's a very interesting, complicated world outside. Jumping out and exploring is important. Many words come around: ignition, innovation, all that will follow. First, let's get out of the well we are trapped in. Thank you very much.
Thank you, Dr. Agarwal, for this encouraging talk and sharing your journey, and significant remarks. So we are sorry to say that Dr. Agarwal has some prior commitments today. So the way we have planned this session, followed by all the speaker sessions, we have a panel discussion planned. So if Dr. Agarwal won't mind, then can we take one or two questions?
Yeah, I'm happy to take questions now. And sorry to my other speakers for disturbing the flow.
No problem, no problem. So I have a question, like, as you said that it's a low hop, not like it's a big jump. So I have a question, like from the panel discussion, topics what we have selected from there only, I'll take one. So when you joined as a, we'll delve into biological research. I mean, what sort of advantage and disadvantages you found in your journey?
So the disadvantage, so let me speak, for example, when I came back to IGIB and worked, I realized I knew so little about areas like microRNA, newer things in genomics, compared to, my understanding of, say, asthma or COPD. And one must realize that there is no problem with this. Collaboration is a beautiful word. You can find people who lack your disadvantages and who appreciate the advantages you bring. And for me, at IGIB, that was Professor Balram Ghosh. And, working together with them, and to the point, it was basically one lab. There was no real separation of my work and your work. It was our work, allowed me to overcome it. And with time, with working with the people, of course, you pick up a bit on that, but some fundamental differences will always remain. And I think nobody can know everything. One must simply be very clear about what one knows, what one wishes to know, and what one simply wishes somebody else who knows will cover for us. And that usually works out well. For example, maths, right? I'm really not inclined to learn maths fundamentally. So anything to do with high-end mathematics, I'll just find somebody who knows it. I have no desire to learn it now.
Thanks, Dr. Agarwal, really encouraging. So on the interest of time, we'll move to our next speaker. Thanks again, Dr. Agarwal, for taking out time from your busy schedule. Now our next speaker is Dr. Namrata Gundia from IISc Bengaluru. She obtained her MSc in physics back in 1994 from the University of Pune, and MS and PhD in mechanical engineering in 2004 from the University of California, Berkeley, USA. After postdoctoral work at the University of California, San Francisco, USA, Namrata joined the Indian Institute of Science, Bengaluru, as an assistant professor in 2008. Dr. Gundia currently in her lab exploring the mechanics of biological materials at scales from individual cells to tissues, especially the structure-function relationships in organisms. So without further delay, let's hear directly from her what she has in her store. Over to you, Dr. Gundia.
Thank you, Sundaram. Thank you also to the organizers for giving me a chance to speak and share my experience. I'm going to share my screen now. Please let me know when you see it.
Yeah, it's visible.
Are you able to see it now?
Yeah, perfect.
Thank you. Thank you.
Wow.
Thank you very much. Thanks to all of you for joining in. I thought I'm going to tell you a little bit about, first of all, my thoughts about, I run a lab which I've called Biomechanics Lab, and that's our logo over here. So as you can see, biomechanics combines both biology as well as mechanics. So I sit truly in the interdisciplinary region, and I not only learn a lot from this experience, but I think I enjoy being in this space. So I want to give you a few thoughts on my ideas of what is biomechanics. I also want to tell you a little bit about how I got here and discuss some of the things like what Anurag has mentioned about the challenges of working in interdisciplinary areas. So biomechanics is not really a new area. So if you think about historically, the number of people, and this is just a short list of my favorite people, mainly because of my interests in mechanics and also and in biology. So we start all the way from Aristotle, of course, but we move on to Galileo Galilei. So this was the Renaissance time, which was a very rich time in science because there were people who were interested in mechanics, like Galileo. And he also began to ask questions of how can we apply these ideas of mechanics into looking at how bones are hollow and treat them as structures like what we see in the world around us. There are others like Robert Hooke, of course, René Descartes, and Newton himself, who was also asking these questions. So I won't go into more detail about all these people, but I would strongly encourage if any of you are interested to go and read about them. But this was a very rich time in our world when the people were able to do all the things that caught their attention. But soon biology became the purview of biologists, and physicists went on to doing more and more work in mechanics and other areas. And this whole enrichment which happened from the Renaissance times was sort of minimized. And we come back much, much later when people again began asking the question of, can we not look at biology from another lens or and things like that. So yeah, so more recently, I think the way in my field of biomechanics, we look at Professor Y.C. Fung, who was at UCSD, as one of the pioneers and the father of modern biomechanics as we know it. And it had to do with advances in theory. Most of these people I've mentioned here are elasticians because that is what I study, elasticity. We also had major technological advances in computing, and we were able to use numerical methods that exploited the computational power, which allowed us to address complex problems in biology. And I do think that with the space race, and which started in the US and now more recently in India with Chandrayaan and Mangalyaan, we are beginning to again see interest in biomechanics and to understand how do our cells and our tissues, how do they handle the kind of loads that they are seeing, and how do they remodel in response to different loads. So I do see that we are back to starting to see this very nice mixing which happens between the fields just because we throw in a few new challenges in our world. My own interest started with looking at, from a mechanics perspective, on looking at metals and ceramics, as most of us are trained. What you see here is stress, which is nothing as force per unit area, versus strain, which is a non-dimensional number. And a lot of people in engineering are interested in understanding these behaviors. And in fact, if you recall, the slope of this region is called nothing else but a Young's modulus, which is something that Hooke had also studied. What you see here is the kind of responses one gets with rubber-like materials, where you don't have a single part which is highly linear. So we have a very non-linear curve which can extend up to 800%. And more interesting, with soft tissues such as skin or with arteries, we see this characteristic J-shaped curve. And again, finding out what the modulus is for this material is very challenging. So I was interested in, can we not apply mechanics into biology, and how are tissues so soft and pliable and yet so tough? Those are the main questions that we think of from an engineering perspective. I was also intrigued with how the structure is very complicated of how we look at things around us. And I've picked specifically an example of an anemone that you see over here. The anemone is another animal which works in the tide pools where things are extremely challenging for the animal. And again, it is very clever in how it needs to adapt to these different challenging environments. The shapes also that you see in biology are a lot more complex than the kind of shapes that we see in engineering structures. So it not only has different boundary conditions, but also very complex layout of different proteins that we see there. And this is a natural aortic valve. And this is part of the aortic valve. So you see the 3 different leaflets here. And this is what happens as we go through disease in some people, and you see a lot of calcification which is deposited. And interestingly, it seems to happen in specific regions of the valve, which got me interested in thinking about how do all these things come together in influencing disease. So this, as I go back through history and I think about how biomechanics has been this very interesting area, I'm reminded again about how Darcy Thompson has influenced much of our thought, which is now over 100 years old. He was the one who talked about how physical forces and internal growth regulates biological form. And this is again a really powerful paradigm that one looks at morphometrics. And we can use this to predict a lot of different ideas. And this is where my lab seems to be heading, is not just in understanding how things are put together, but also can we come up with models to understand how they grow, or what is the contribution of growth in the way we maintain our 5 fingers without adding more fingers as we grow older. So going back to a little bit on how I've come over here. I started with bachelor's in physics and master's. I always thought I liked physics, and I never wanted to do engineering when I was in India. I started with a PhD in ISC, but then I quit and I moved to the US. And it was in the US that I decided to change and got and started thinking about what I really wanted to do with my career. And I got interested in biology. And I do think that at the time when I joined, which was in 1998, a long time ago, I think US is one of the places where it allows us to make these transitions much more easier than what we can in India, although we are changing as we see. So I joined a lab which was studying biomechanics, and that is the reason I changed into mechanical engineering and didn't stay with the physics training. But I see all my training start to come together as I move through finishing my master's and PhD, and then my postdoc. And as I joined my lab and started one in ISC, I see how my interests in physics and engineering have all come together to frame the kind of questions we pose. I'll give you a brief idea of the kind of things we do in my lab. So my lab, again, like I mentioned, is highly interdisciplinary. We work at the interface of mechanics and biology. We also collaborate a lot with clinicians because much of my interest lies in understanding disease from the perspective of mechanics. We also work with some companies so we can design methods or tools to mitigate some of the problems that can come with disease. So my main interest started in a rubber-like protein called elastin, and I studied a lot of its role in arteries and how the artery is a nice composite heterogeneous structure made up of these different kinds of proteins which are put together, and how they influence the overall properties of the arteries, which allows them to extend up to 80% during a given cycle and go on for at least 85 plus years of our life without really seeing as many problems. But of course, as we know that we do get atherosclerotic plaques, and there are also places where people get, the left ventricle, which I'm showing here, is also a place where sometimes we get these transmural infarcts, which is something that I studied during my postdoc. And here, what you see a lot of patients in India also go through this is the cardiac cells die, which is shown in this black region. And what is again interesting is this whole transverse section that I'm showing in this picture starts changing. This region gets thinned out, and the whole idea of what I call as growth, which you might have thought of in a very positive way, is now again reflected in how this region is growing, but not in such a good way, because this patient is going to head into diastolic heart failure. And if they survive these transmural, this myocardial infarction. So my first project as a postdoc was to look at how can we assess a device, which is essentially a sock that is put around the heart. And the idea was that the sock would prevent the heart from bulging out. And so the question I asked was that how can we assess the influence of this as far as the heart mechanics is concerned? So what we did is we used again methods in engineering where we discretized the left ventricle, which you see in this picture over here, into little cubes. And we look at properties at each of these, which I will call nodes. And we assessed the pressure volume relationships, which give us an idea of how the heart function works. This is a technique called finite element models for some of you who are interested. And it allows us to put various kinds of boundary conditions, which is again a very engineering tool that everyone in engineering is used to. What it allowed us to do was to pose the question of what happens as soon as a person has a myocardial infarction, when the tissue remodels from somewhere in the intermediate region, but it goes towards this chronic condition, which is shown in red. And you'll also see this diastolic region, the diastolic part of this curve is again getting dramatically changed. So we were able to make all these predictions and work with clinicians to assess the contribution of this sock-like structure and how it influences heart function. So in my lab, it was challenging to set up connections with the clinicians when I came back in India and started my lab in 2008. So what I decided to do was also to address how do cells contribute to these properties of the tissue. So what we looked at was how do cells adhere. I also work with, how do cells adhere to substrates, how do they migrate, and how do they contribute to this changing mechanical environment during growth. So I work on understanding again the aorta, which is another region of interest, where we have developed experimental models to study flows through arteries. And we also use numerical simulations to see how these flows separate out in this region. And you'll see these vertical structures which have developed. And how do these vertical structures influence cell mechanobiology? So here, two students from, one is from another colleague's lab and Neha, who's in my lab. We look at using microfluidics and we expose endothelial cells to these bidirectional stimuli, and we watch the shape and we try and understand the function of these endothelial cells. We also are excited about how cells adhere. And these are again methods in mechanics that we like to use. So these are classic problems which are called inverse problems, where one gives a displacement field and one asks the question, can I now predict how much amount of stress is being exerted? And so what we do is we show how our methods can show very nicely the form and the adhesion, which you see very nicely in this picture, this is a boundary of a cell which is adhered to a very soft hydrogel, and we can show the kind of nodal force vectors and how the cell is pulling on the substrate as it adheres. We also design microfluidic, micro-patterned substrates. So these are about 3 micron and 6 micron height. And this allows us to watch how cells move. And you'll see in, so these are lines that we have created, and like very good, car drivers who stick to their lane discipline, 2 cells, one on the north and one over here, are migrating exactly along these tracks, unlike some of the drivers in Bangalore, at least. So this allows us to not only watch the adhesions, but it allows us to quantify how much traction cells exert as they adhere. So again, these are all problems that we have in mechanics, which we have applied into biology. We also build devices, again, a very mechanical engineering heavy thing. So we build devices to subject cells to shear, and we can watch how cells de-adhere from substrates. So this is another area which is like mechanodiagnostic. So can we use such ideas to understand what is a cancer-like cell and what is not a cancer-like cell? And can we look at its de-adhesion characteristics? So some of this work is in collaboration with 2 friends, Gautam Menon and Pramod Pularkat. We also build devices to subject cells to differential types of stretching, and we're able to watch the cells during the de-adhesion, which is another thing that really is something that we really like. We are also interested in to understand how they migrate, not just how individual cells move, but you'll see that cells are drawing from various kinds of stimuli and they interact with their neighbor particles. But you'll see one of the cells here, which has broken up from the rest of the cells. And it has, so this is not exactly what I would like to call a leader cell. It has walked away from the rest of the population. So we want to understand how do these different cues, which again influence how cells move, which comes back into where my initial training was, which was in physics. So together, all of these thoughts of how mechanics and biology contribute to how biology works is a central question which is of interest. This is my current lab, but I just want to add a few thoughts on some of the questions that I was asked to discuss, which is what are the challenges I face working in this area. So it is very exciting to be in this area because the questions that I pose are mainly come from biology, but the entire techniques that I throw at understanding these questions come purely from mechanics, whether we call it engineering or we call it pure physics. So a lot of it comes from mechanics. This brings problems, and because some of the things that one wants to do is to be sufficiently, you have to be sufficiently wide in being able to talk to the different people who are your collaborators. So I need to be able to talk to clinicians in a language that both of us understand each other, and to biologists in a language that we are both familiar with. So it requires a lot of training. So I did have a minor in bioengineering during my graduate school, but a lot of time is spent in trying to bridge this divide. I'm also constantly challenged because this is a new field, relatively speaking, in India. So me being comfortable in a department which is purely mechanical engineering, and to be able to convince your colleagues that this is not biology, but this is biomechanics, also becomes important. At the same time, I also have to tell biologists that I'm not a biologist, but I use techniques in mechanics to understand these questions. So those are some of the challenges I face. The students also come with their own set of biases, especially engineering students, because the engineering students don't normally think of doing biology. So it takes a different kind of person to appreciate this problem, and it takes a different kind of training for someone to be more accepting and to make more innovations in this area. I think I'll stop there with my thoughts, and maybe I will, if you have any questions, I'm happy to take it.
Thank you, Dr. Gundia, for this amazing talk. And you nicely sandwich your talk between your career trajectory and your exciting research. Thanks for that. On the interest of time, we'll move on to our next speaker, and we'll be moderating all the questions later in our panel discussion session.
Okay, thank you.
Thank you again. So our next speaker in today's session is Dr. Jayathi Sangupta from CSIR Institute of Indian Institute of Chemical Biology, Kolkata. She had a background in chemistry, and she got trained as a PhD student at NEHU, Shillong, under the guidance of a chemist, Dr. Richard Duncan. And after that, she spent a short stint as a DBT postdoctoral fellow at Bose Institute, Kolkata, on molecular modeling and structural analysis of peptides with Dr. Gautam Basu, before joining the laboratory of Professor Joachim Frank, incidentally, who won a Nobel Prize in chemistry in 2017 at Wadsworth Center, Albany, New York, USA. Currently, in her lab at CSIR IICB, she combines structural biology with functional studies of higher order biological complexes to understand their roles in health and disease. So it's over to you, Dr. Sengupta. And I'd like to mention that despite her troubled health, she joined us, and we truly appreciate her effort and commitment. Without further delay, over to you, Dr. Sengupta.
Thank you. It's my privilege to get this chance to share the virtual stage with all these eminent scientists. And I thank you all, the organizers, for giving me this chance. I apologize for my voice. Hope you can. And I'm sharing my screen now.
Yeah, it's visible.
You all can see, right?
Yeah, good to go.
Yeah. So basically, I am going to tell you, I can take you through my journey down the memory lane. I go how from chemistry I came across to biology, and so it's a kind of a story I'll tell you, and some of you may find it very similar, or you may not, because it was a pre-internet social media time, and we particularly being a suburban town student, we didn't have any idea about interdisciplinary sciences and streams, and probably there was not much options also. So I started as a chemistry student. I had an impression that physics is too abstract, and biology with all these dissections and all, it's too real. So chemistry is in between, and I started studying chemistry. And then I moved for master's to IIT Kanpur. Again, chemistry was my subject, and I had more like preference to inorganic chemistry, particularly coordination chemistry, and we didn't study anything biology till then. And the fourth semester, we had to do a project work, and I started working in Professor Parimal Varadwaj's lab. And I had no idea that inorganic chemistry has any overlap with biology, but in this lab, I came to know that his supramolecular chemistry lab, that time he was trying to make some metalloenzymes mimic. And I came to know that biology uses a very nice trick of chemistry, like copper 1, if I remember very correctly, copper 1 prefers tetrahedral geometry, and copper 2 prefers a square planar geometry. And these enzymes during this redox changes, because of this preference of the ions, metal ions, the protein changes its conformation. So it was quite fascinating. But still, I didn't have much idea or to chemistry. And then I moved to Shillong, Nehu, for PhD. Many people ask me why I moved from IIT Kanpur to Shillong for PhD, and probably my answer never sounds very convincing, unless someone really sees Shillong in November. It's a clear blue sky and flowers everywhere, and you could see basically Van Gogh's Starry Night on the nature's canvas. So it was really so I liked it so much, so I stayed there for PhD work. Of course, probably it was not a right decision, or I don't know. Such a nice nature really distracts you from studies. But I studied DNA modifications by polycyclic aromatic amines. And it was chemistry, but my focus was to know how the DNA bases are changed. But during that time, I had to know the parameters of DNA, how the DNA looks, and how the base pairing occurs, and sugar puckering, and all this. So I had some exposure to biological polymers. Then I came to Roche Institute, transition to protein, from DNA to protein. I studied protein structure, peptide structures by NMR, and I started getting an fascination on structural biology from there. So till then, it was really not much idea about the biology as whole, but through chemistry, biology started coming into picture, and structural biology, I started having a preference. And after say about 2 years, I moved to Professor Joachim Frank's lab. It was in Albany, New York, State Department of Health. He was there that time. it was a ribosome research lab, and he used to, he still uses a cryoelectron microscopy. He was one of the pioneers who developed the method, cryoelectron microscopy. And frankly speaking, I didn't have any idea about ribosome, except that it's a molecular machine that synthesizes protein, and neither about cryoelectron microscopy. And in his lab, I met the astounding molecular machine, ribosome. And it was, I can say, love at first sight. It's a atomic scale, you can say it's a huge macromolecule, as you all know. It's a RNA protein complex. And the time I joined this lab, his lab, it was very, that year 2000 was very important for ribosome research. All these crystal structures of ribosome came out that year, first came out, like Venki's structure, 30, a smaller subunit structure came, and Ed Ionat's and Tom Stey's group published the large subunit structure. And from Joachim Frank's lab, that time cryoelectron microscopy was limited by my resolution. So it was the first pretty good resolution map was 10 angstrom, and it was published in Cell. So the atomic structures of the small and large subunits were available, and there was a need to make a molecular model of the 70A structure which Joachim Frank's lab made. So I was assigned for that project. And as I said, the ribosome structure was limited by resolution, means you cannot resolve very small changes through these maps. And nowadays, you know that cryoelectron microscopy has reached this resolution revolution occur. Cryo-EM can reach near atomic resolution structure, and that days it was kind of a blobby kind of structure, and now people really make fun that it was a blobology. But I can just give you a glimpses of even that blob structure, some very important structural changes we could find. So one example I can just show you, you all. This was elongation factor TU, which delivers the tRNA to ribosome. So this there was a long-standing question how the tRNA, the way it accesses ribosome, it is difficult to get paired with the mRNA. The geometry is not like proper. So, and that time it was, the idea was the tRNA is a very rigid kind of molecule. So the first structure, it was about 10 to 12 angstrom structure of EFTU bound ribosome. It was seen that the tRNA is actually a bent structure at the anticodon end, and it actually, to read the mRNA, you can see that you can pretty good, nicely model this tRNA here. And this kind of bending is essential to read the mRNA codon. So the anticodon actually bends. tRNA has that flexibility. So this structure was about even 10 to 12 angstrom, it was determined. And another conformational changes, very, very big changes, which could be identified from the cryo-EM structures, that it was elongation factor G bound structure. And you can see the small subunit actually rotates over the large subunit like a ratchet kind of motion. So these maps were about 10 to 12 angstrom, but the molecular model built on it could really determine the conformational changes. And now all those recent near atomic structures, they show the same, I mean, it confirmed that even though the resolution was very low in that what was identified, the conformational changes are actually correct. And then another very interesting changes also was identified. It is a release factor. So the release factor, which actually releases the polypeptide chain from the ribosome, the crystal structure, the isolated crystal structure, actually shows a compact kind of structure. You can see, if you can see my cursor. So this is a very compact kind of structure, but the biochemical, biophysical previous data showed or identified two motifs. So this release factor, it needs to read the stop codon, and then it needs to cut the polypeptide chain. So it actually should reach from the anticodon to the polypeptide chain, the site where the polypeptide chain is. That means like the tRNA, it
Happy
should have
birthday!
Hello?
I'm sorry for the interruption. Please go ahead. It was
okay. So these two motifs should be actually like the tRNA, should be about 75 angstrom apart. But the isolated protein crystal structure showed that it is only 25 angstrom. So it was really not clear how this protein actually functions as release factor. And then in the cryo-EM density map, which is again about 10 to 12 angstrom resolution, it was found that these particular domain actually moves far away. I mean, it just changes its conformation and reaches to the CCA end where the polypeptide chain is located. And these kind of big conformational changes occur after binding to the ribosome. So these are few examples which was the work was done in his lab during that time. And now the cryoelectron microscopy, as you probably all know, that it has reached it has the capability to reach atomic resolution structure, and chemistry is required to analyze a atomic resolution map. So it is actually the problem in addressing by chemistry is actually the role of chemistry, as the previous speaker also says. The problem, biology is a gold mine of mysteries, and to address those questions, they're all fascinating questions, and any background you can address the question. And whether this chemistry background, the question is whether it has any difficulties, or yes, of course, because I never studied biology, so the knowledge that already came to the textbook, I didn't have the chance to go through that. I often need to probably go back to textbook if needed. But again, the questions, addressing the question, it can be different levels of understanding. And this is a sentence that my supervisor added in my thesis, that there are different levels of understanding a problem. It could be histological, it could be cytological, but I had this question to address a question at molecular level. That is it drives me. So that is because probably the chemistry background that makes me to see problem to solve at molecular level. So this is my experience, and in our lab, we study ribosome primarily with cryo-EM as a primary tool, and these are a few publications. I don't want to go through the problems, so as I said, the biology is a gold mine for problems. It's so much is known, but still it's started with mystery. So many questions. So as you say, all roads lead to biology. So yeah, I want to stop here. Thank you all.
Thank you, Dr. Sangupta, for this wonderful talk and sharing your nice career trajectory and your exciting research with us. So on interest of time, we'll be taking questions later in the panel discussion session. So next, moving on to our next speaker. He's Professor Sumantra Chatterjee from NCBS, and popularly he is known as Shona Chatterjee, as you all know. Currently he is at NCBS Bengaluru, and he's placed in a very distinguished position in India, and he's the first Indian neuroscientist who had been elected as a lifetime member of the EMBO for his outstanding contribution in neuroscience. In his lab, he's busy with studying the cellular mechanisms underlying the emotions using a range of behavioral techniques, morphometric, in vitro and in vivo electrophysiological tools to answer all the mysterious questions of neurosciences. So over to you, Professor Chatterjee.
Can you hear me?
Yeah, it's perfect.
And I'll go into see my slides as well. Can you see my slides?
It's visible, yeah. Great.
Okay, so I'm going to go ahead and start my talk. First, thank the organizers of Scimplify for this really interesting venture and for allowing me to participate in it. I will talk about my own experience and journey like the previous speakers have. And the title of my talk is Following My Bliss, which I have borrowed from one of my favorite quotations from Joseph Campbell, who's an expert in mythology. And the quote runs as follows: follow your bliss. If you do follow your bliss, you put yourself on a kind of track that has been there all the while, waiting for you, and the life you ought to be living is the one you are living. And I'll give you some examples from my own experience to make the case for this. So briefly, I'll talk about where I'm from, what kind of choices I had to make, the transitions that entailed making these choices, and some lessons learned and applied. I hail from Shantiniketan, and I studied in Tagore's school, Patho Bhavan. I'm actually a third generation from both sides of my family from Shantiniketan, so it runs very deep in my veins. It's a wonderful environment and wonderful experience. Grew up immersed in nature, and the school was unique in the sense that it imposed no hierarchy of subjects. All subjects were treated equally. If anything, there was a greater emphasis on liberal arts, music, fine arts, and so on. I had equal weightage of marks in my final year in school for mathematics and music and art. One of the early influences in my life, which was when I was in grade 2, was that I was allowed to stay up all night and watch television to see the Apollo 11 moon landing live on TV in the US. And that had a very deep impact on me in terms of loving physics and especially space science. Once I got back to Shantiniketan, there was a countervailing force, which was the life in Shantiniketan itself allowed me, without consciously realizing it, that I was learning from my surrounding environment all the time. I was immersed in nature. All my 5 senses were getting stimulated all the time. And in some subliminal way, I was picking up biology without realizing it, although I really hated the subject in school. Another big influence was Germany, where I went to school for grade 6 and 7, where a lot of things were taught hands-on, including biology and geography and so on. There are 3 books, 3 sets of books that really had a deep impact by the time I reached grade 7 or 8. Was Ascent of Man, Jacob Bronowski's book, and the documentary based on it. The Double Helix by Jim Watson, the story of the DNA, the discovery of the DNA structure, and several books by Carl Sagan, such as Crocus Brain, and so on. And so these were tilting me towards biology, especially brain science, by the time I was in grade 10. So I was sort of faced with a dilemma which got sharper when I finished high school in terms of life sciences, which I personally liked very much reading, but it was taught in a way where you had to memorize everything. So I had put it in the same traumatic folder where I had parked chemistry and Sanskrit, because you had to memorize everything. And on the other side, I loved reading physics and learning about physics. Feynman lectures were fantastic to read. And so I had made a choice when I was applying for college. And the applications I made for college were in the following order. I applied to the history department, the School of Fine Arts, and then decided that I couldn't make up my mind and needed to buy time, and hence got into physics so that it would just give me a little more time to think about stuff I like, to be able to follow and find my bliss, so to speak. And that eventually led me to, after finishing my undergraduate, to go and do a master's in physics in IIT Kanpur, which was a star-studded place in those days. Very, very bright students and brilliant teachers. And I really saw the kind of bright minds that were going to impact physics in a fundamental way in the early days. And at the same time, my interest in biology grew stronger, although I had no formal access to anything in biology at that time. But slowly I realized that I was more attracted to hands-on, kind of messy experiments where I can touch and feel things and have an organic sense of the science, versus the kind of physics that was being championed at that time in those departments amongst the very, very bright students was theoretical particle physics and things that felt a little remote to me. So as a result, I decided to take the first major plunge and high-risk transition, which was to abandon physics. And I could have possibly, like most of the students from IIT Kanpur in those days, walked into any other graduate program in physics in the US, but decided against it and only applied to biology and biophysics programs in the US, and managed to get a fellowship at Johns Hopkins University, which was a sort of a lifelong dream of mine to go and study there. And that allowed me to enter a department which was small, which I could get a lot of attention, to make that tough transition from physics to biology, because I had really no formal training in biology at that level. And so this is where I started pivoting to neuroscience through a whole bunch of crash courses in biophysics, neuroscience in the medical school, summer courses in the marine biology lab in Woodsole, and so on. And all of this was enabled by the fact that I was very fortunate to be accepted in Terry Sinowski's lab when he was in Hopkins. And he was a pioneer in computational neuroscience and neural networks. And he himself had already made that transition a generation ago. He started with John Wheeler and then John Hopfield in physics, and then moved into neuroscience with some luminaries in the field. So he was used to this, he liked this, he encouraged this, and his lab was filled with examples of role reversal, meaning biology graduates were doing computational models, theoretical models, and physics, computer science, electrical engineering graduates were doing neuroscience and messy biology. So the bottom line was there were no dogmas in the lab. There were nobody who was an expert in the field. Nobody had a clue what they were doing, but there were all a bunch of bright minds who were chasing really cool questions. So it had a sort of a Wild West kind of flavor to it, which is a perfect landing spot for somebody like me, who was really clueless about biology at that time and needed some time to figure things out. And so this was a very nourishing environment. But just as I was settled down in Hopkins, something interesting happened, a little challenging at the time. Terry decided to move to the Salk Institute, which did not have a PhD program and did not encourage graduate students to come and join. But I tagged along in the hope that I'll survive. And I picked a PhD question, which was again embodying a sort of a paradigm shift and a transition, which was to test one of the predictions from Terry's neural networks research, a theoretical prediction, and translate that into testing experimentally whether they apply to synaptic physiology and plasticity in the hippocampus, an area involved in learning and memory. So this is the work I started there. Salk was, again, a sort of an amazing place, an amazing ecosystem, in addition to its stunning natural setting shown here on the right. It was also a stunning place in terms of its intellectual ecosystem. I did not have any formal coursework there. I had no structured curriculum. And I was surrounded mostly in the lab by theoreticians and postdocs, no graduate students. But it was an amazing intellectual ecosystem. The lab used to have a tea at 3:30 every afternoon, hosted by Francis Crick. And he would invite anybody who's around, including Ramachandran, Pat Churchland, John Stevens, Jonas Salk, shown here on the right, would roam around the courtyard wearing his lab coat, and he would pop in, and many others like this. So I essentially learned a lot around the tea table, more than anything else, and in the lab. And all of this was a once in a lifetime kind of experience. And then I transitioned to my postdoc and in Yale, and I decided to do something completely different. I was trained in a computational neuroscience lab, and I walked away from that to focus on an area of the brain that was quite understudied and unknown at the time, called the amygdala, which is the hub of emotional information processing in the brain, which is involved in forming fear memories, for example, and involved in a whole bunch of disorders of emotions. And it is during this postdoc at Yale that I had a very, very important personal meeting with Professor Obaid Siddiqui from TIFR, who was in the process of establishing NCBS in Bangalore. And I had multiple long personal discussions with him in person in Yale and later in New York, where I got to learn about his vision for NCBS. And by that time, it was very clear that, you have one long lunch with NCBS, with Obaid, and you're sold on everything. And that's what happened to me. I decided to come to NCBS, although it really didn't exist in its current form at that time. And so I had to buy some time because NCBS was still getting constructed. The campus was not ready. The labs were not ready. So I moved to MIT sort of as a placeholder for sort of a holding pattern for a while till NCBS got ready. I also got to meet many of the pioneers of NCBS at that time in MIT. So all of this time was spent not only preparing for the future at NCBS, but also starting thinking about entirely different kinds of research questions that I had not explored during my training earlier. So the final sort of transition, the fourth transition, if you will, was coming back home and starting my own lab at NCBS. One must realize that that NCBS was not today's NCBS. It was still very much a work in progress. A lot had to be built. A lot has been built by the pioneers, but more had to be done. So it had a kind of an open-ended frontier mentality, which was quite liberating because it gave us lots of freedom to try out stuff and fail, and it didn't matter. But it's a very exciting time to be in NCBS in those days. Now, with that freedom and that very rich environment that Obaid and others had created, I decided to take on something that I'd never done before, completely outside my domain of training. And there are two questions that we decided to chase in the lab. Why are emotional memories so much more robust and long-lasting compared to memories of facts and events? You can appreciate that from your own experience in your own life. You can have one very happy or very traumatic experience that you'll remember for life, yet memories of daily facts and events, stuff that you need every day, often fade very fast. And why is that? Is one of the questions we've been chasing in the lab. And a question that emerging from that bigger question is that stress, behavioral stress in life, also happens to have an opposite effect. Stress enhances emotional output in the brain, yet in the same brain, it impairs factual memories and cognition in general. So why is it that the same top-down perturbation in the form of stress has completely opposite effects on these two types of memories? Those are the two questions that I decided to pursue in the lab, although I must warn you again that I never worked with animal models before that. I had no experience in that. And I had no experience in neurobiology of stress, fear, or anxiety at all. So again, diving into the deep end of the pool without knowing what the hell I was getting into. And I did that by taking something, I guess, which is our engineers and physicists are quite used to, is a reductionist approach. You pick a problem at one level, make approximations, and you work your way down the ladder and dive into more greater and greater detail. Those details that emerge give you predictions that you can then test at other levels of biological scale, either above or below. And you keep going back and forth to get a view at multiple levels of neural organization that together give you a picture of what's going on. And this is, I think, an approach that has worked well for us, and this is what I've used throughout my work in NCBS. But I think it has some of its roots in the way I learned to think about problems from physics. I'm not going to go into any further details of the science here, but rather I'm going to just talk about lessons learned in this whole amazing journey. The first very humbling experience, of course, was, and a realization first, early on in Johns Hopkins as a graduate student, was that this ain't physics. There were no Newton's laws or Maxwell's equations to guide you out of trouble. It was a really messy system that we're studying. So you need a lot of luck, and you need good mentors who believe in you to back you up in the dark days when nothing worked. The other thing I think probably came handy from physics is that physicists are pretty courageous in making approximations. You're faced with a very complex problem and complex system. You don't get bogged down. You figure out, you ignore some facts, take on others, make some rough approximations, and get started. And that is something very useful in taking on a new area of research such as neuroscience. And so in the process, what I learned really is learning to learn, and also knowing what I don't know, so that I can go back and seek help from others, something that Dr. Agarwal mentioned in his talk. And also, as a result, because you have to depend on others and other expertise, experimental biology is we, not me. You really have to be collaborative by necessity, not by style, just not as a fashion statement, but by necessity. And of course, something that I faced along the way, and all my students and postdocs face all the time, and it's true for biology in general, is that try again. And even after you try many times, sometimes nothing works. And that's when you go and change your socks and hope for the best. So this is again a very humbling experience in terms of switching from physics to biology, because you cannot rationalize a so-called failed result or a negative result or a positive result. It just works. And if it works, stick with it. If it doesn't work, don't take it personally. And of course, all of these are challenging issues that one has to deal with. So it becomes worthwhile going through all of this pain and uncertainty if you're chasing a cool question. So a nose for cool questions is important so that it keeps you going when things are not working. How have I applied these lessons in my own lab in NCDS? Well, because as I told you, I started way outside the box. So I'm quite comfortable outside the box. So I've never hesitated to stay or wander outside the box, and I quite like it. There are, of course, risks of not knowing every biological detail, and sometimes it catches up with you. But it is also fun to be outside the box because you're not bogged down by dogma, and you can try things out, and it's boom or bust. Sometimes it works, most of the time it doesn't, but there's never a dull moment. And this is something I have thoroughly enjoyed in my journey, and I always try to convey this to my students. In terms of my own lab, the few lessons that I have imbibed from my mentors in various places, I've tried very hard to apply them to my own lab. So I fill my lab with folks with a wide range of backgrounds. My first graduate student came from agriculture, and subsequently I had students from physics, computer science, electrical engineering, along with kids who were trained in traditional disciplines of biology, physiology, microbiology, and so on, and in medicine, because they know their stuff better than I do. So it's a combination of these different pools of talents that I'd like to throw in together in one space and then see what happens. And always try to find talented people and empower them. And then once you've picked your horse, back them 200%. And in the process also, as you find these very gifted people, and there are many, many gifted people in India who are willing and eager to do neuroscience, is to encourage these same transitions that I've lived through, encourage and enable the same transitions in their lives as well. Because there's hesitation in this system, because the different disciplines have major barriers between transition from one discipline to another, and it's not encouraged. So I always try to give them the courage to do so and jump off the deep end of the pool, just like I did myself, and tell them it's okay to try. And so that's I think worked out rather well. And I'd like to end here in the interest of time and just hope that the story I've told you is useful and interesting and adds value to what you have heard from the previous three speakers. Let's see, the slides getting stuck. So I'd like to thank my students, postdocs, and colleagues, collaborators, and mentors who have helped me in this journey all along over the last 30 years or so. And I'm very grateful to all of them for their help and advice. And I'll end by quoting Jimmy Buffett. That's my story, and I'm sticking to it. Thank you.
Try. Excellent, amazing talk, Professor Chatterjee, and for sharing this exciting and very, very exciting career trajectory and motivating us. With this, we'll move on to our panel discussion session. We'll request all our speakers to kindly switch on your video so that we can see each other and we can start out. Maybe Dr. Sangupta, I don't know. I mean, she had some troubled health. If you are, no. Thank you.
Can you see me?
Yeah, great. Always great.
Okay, so I'm just gonna. Is my thing still visible annoyingly?
Yeah, it's visible. Yeah.
Right.
Can you please stop sharing?
It's going to a funny mode here. You know what? I'm just going to log out and come back in. Okay, there's something stuck here. All right. Yeah.
Sure. No problem.
You guys go ahead. Yeah.
Okay, thank you. Once again, thank you all the speakers for sharing their motivating story and inspire us. So with this, we'll move on to. maybe Dr. Agarwal won't be able to join. He has some prior commitments. Dr. Sangupta and Professor Chatterjee. Let's wait a bit. Professor Chatterjee is joining. And it's great to see that chat box is full of questions. Excellent. Thank you all for being so interactive. And again, thanks speakers for their time. Hello, Professor Chatterjee.
Oh, sorry. Yeah. Is there a question for me? Sorry, I missed it. Yeah.
No problem. No problem.
What's the question? Is there a question for me?
Yeah, so, before we start, can you please switch on your video? If not, a problem.
Did switch on my video? Or switch off or switch off on?
Switch on, please. Please switch on.
It is switched on. It is switched on.
We are not able to. Yeah, now it's perfect.
Okay.
Yeah.
All right. Yeah.
I can see. Great. So before moving on to the questions from our audience, we'll discuss a little bit on a few of the questions what we have selected for the panel discussion. So these are the questions to all the speakers, and just take your time and whosoever start, just start, and followed by we'll be moving on to our next questions. So let's start with the first question. What we have is, how have you been motivated to switch your gear to your biological research from apparently a different domain of expertise? What was that drive which motivated you? Can you please share with us?
Well, I can go first since I just spoke most recently. I think there was no one sort of aha moment except for a gradual realization that some of the most exciting work in science was being done in biological sciences, and neuroscience being at the forefront of that. So it was an easy sort of attractive sort of feature of neuroscience that attracted a lot of people from different areas actually. And so it was an easy choice in some sense, except that making the transition was not very easy in India at that time. And it was a lot easier when you went abroad, because that was already permissive and was already happening. It's now happening in India also, by the way. So, but I think you know, these kinds of different disciplines of science come in waves, and you know, the tide comes and goes. So it's a matter of having an awareness of what's picking up, but more than that, it's really what grabs you in a fundamental sense. What kind of questions grab you, and that depends on who you are, how you trained yourself to look out for questions. So I think it's a very personal decision in some sense, but having exposure, wide exposure, and having your eyes and ears open to the possibilities, and being in a system that allows those possibilities to come to you, I think is very important. And so it's partly individual and partly a matter of the educational system. Both have to play a synergistic role to make that happen.
I can go next, Sundara. Is it okay? Yeah. So I agree with Shona.
Yeah.
So I agree with Shona that it is a personal decision. Also, I think we did not have that kind of exposure as much when we were young students in India. I think a lot of such forum and more exposure to talks from interdisciplinary areas are always exciting. And I didn't hear as much of it when I was growing up as this. But I think it is easier now also, like Shona said, I also felt the transition was easier in the US because the system there was more mature to accepting people to transition into different areas, whereas we were a little more stuck in our old ways. And I still see that, unfortunately, a lot in the engineering sciences, where students are not as much adventurous in moving into different areas as compared to, I would like to say, physicists and also biologists. I find physicists very open to embracing change, and I feel biologists are very open to welcoming people from various disciplines into their traditional areas. Both of these have to happen before we can foment these kind of ideas. And I think now we're starting to see change in the Indian ecosystem. It wasn't the case when I was growing up. So I think for me also, all of this exposure certainly helped. And as a young student, my father, who was also a scientist, would always bring us books on, time life series and, exposure to areas of even watching documentaries like Carl Sagan's, like what Shona mentioned, of Cosmos. All of these are essentially important for us to embrace change. So those are some of the things which influenced my decision into going into biology, because questions are so rich in biology. Thank you.
I should also add that institutionally now, the ICERs have embraced a different model where they allow basic common curriculum of undergraduate education before you start picking your thing. And that's essentially an undergraduate model from the US, which I think is excellent that India has done this. There are multiple ICERs. IISc has started an undergraduate program, which is outstanding. They attract some of the best minds. I would say those minds are better than many of the IIT kids now. And I mention this because let's not paint a rosy picture. A generation ago, just like Namrata did, Namrata's a lot younger than me, is that in IIT Kanpur, for example, once my professors got a whiff of the fact that I was going to walk away from physics, some of them came at me quite hard because it was violating a framework and a dogma. And I was laughed at, scoffed at dinner by some professors who said, oh, biophysics is for those who cannot deal with Schrodinger's equation. So you're getting slammed because of this stuff. And that's not easy for a young person to deal with, right? Because the proven course is to stick with it, go to a good place and do physics. And so there are individuals at that time, not from the physics department, but from the chemistry department, where it was far more accepted that chemistry graduates would go into biology. All my classmates in chemistry actually went to biology. And I stuck around with them and the chemistry professors to get encouragement to go into biology. So, these structural changes are there, barriers are there, but I think they're a lot lower now, already in ICERs as well as in the various research institutes.
Thanks for sharing, really insightful. Now, Dr. Sangupta, can you please unmute yourself? You are muted now.
Yeah, I'd say that biology kind of differentially integrated in the path. So I still feel it's chemistry. I mean, it's why not? It's a chemistry. It's only the problem is in biology, and same to other fields, I think. It's the question you are addressing that is from biology. And the inspiration, if you say, as I told in my talk also, that I just, it came like that when I did this project in bioinorganic. I didn't have that idea that biology has, inorganic chemistry has anything to do with biology. But then I found that it's a simple copper changes of its state, it can change the conformation of so the structural biology came into I started getting interested from then, and still I probably didn't think of going into biology. I was still in the chemistry. I thought that I'm learning chemistry, but only the problem is protein or ribosome now. It's a big complex. So Now it's, so many, like students are now exposed. In our time, as I said, we had no single course in biochemistry or anything. It was, so we didn't have any textbook knowledge that way. But now students are exposed. So if they find biological questions very intriguing, they can come. Back then it was not there. So it just, for some people, it just, you came across and you started loving it. And so that's how it came for me too. Yeah.
Amazing. So in line with the question, I think two of the speakers talked about the changes what happened in the recent times in the Indian education system. So in this line, do you feel that our system current of what we are having is up to the mark, or we need to evolve more to embrace the, the more interdisciplinary nature of science? What is your view?
Namrata, you go first.
Yes, so the change is obvious in every system, so I don't have a concern about that. My concerns about interdisciplinary work are not, so one, we are getting into a lot of exposure into these different areas, but I still have to caution that whilst we move into interdisciplinary areas, we have to be well grounded in one of the disciplines, which is a very big challenge, feels like most of ours faces, is I identify as a mechanician who does problems in biology. So the challenge is to have that depth in a subject area, and be conversant with the cool questions, what Shona called, in other areas which come about. So yes, exposure is required. So, forums such as this one and other talks that you hear. I'll give you one example. In my own department, I find students are very reticent to hear talks in various subject areas. Unless and until it is almost like one of your own lab talks, they're thinking, how is it relevant to my area of work? This is not how things work. Because this cross-pollination can only happen if you talk to more scientists in your field and you listen to more talks. So I feel that we face both challenges in our system, not just to open up and be interdisciplinary, but to also be sufficiently well-grounded in our individual disciplines.
Yeah, I think it's a very, very important point in terms of what do you bring to the table. A student can be trained in any subject, but they have to bring something, core capabilities and intellectual framework that they're good at. And so interdisciplinary is a catchphrase which is slippery in the sense that you don't want to be somebody who's a jack of all trades and master of none. It has to be the opposite way around. You have to be master of something, and then you can go out and explore other trades. And the question I often get from incoming students, especially in the older days in NCBS, for example, was that, oh, I'm not sure I can do neuroscience. I know nothing about neuroscience. Can I do neuroscience? Was a sort of an existential anxiety-driven question that I used to get early on in the first rotation. And all I told them is that everybody can do neuroscience. That's not the problem. What are you good at doing? What do you want to do? So, you've got to find out what you want to do is a double-edged sword, right? You have to first know what you're interested in, of course, and then you also have to find out what you're good at, right? Your hands, if you're using your hands, you're certainly good at some things and not other things. For example, some people are very comfortable doing behavioral neuroscience with animals, others are very uncomfortable, right? So there's an issue of interest and question and what inspires you and what you actually can translate that into in terms of your own core skills and capabilities. And so there's Namrata said that realization of these two things, the earlier it comes, the better. The sooner you get on track with that and understand that, it's better. And there again, I think the flexibility in the education system would be good. And all I will say here, at the risk of becoming a little unpopular, is that our traditional teaching in biology still remains largely a very long, ever-growing list of laundry list of facts. right? And that you have to memorize in school, as opposed to the basic math, physics, chemistry in this country at school is taught rather well. They learn quite a bit and they're very good at it. The teaching is good. The teaching is mature, has a long history. So there's this balance, I would say. On the one hand, you're memorizing stuff and catching up with buzzwords. And the other is you're essentially learning to solve certain problems and getting core skills. And so they're two very different worlds. And how to integrate them in a way in the education system early on is a big challenge, where the research institutes that we work in contribute nothing to that process. And we are guilty of that, right? We are not really feeding back into the education system that we're complaining about, right? Yet we expect them to produce students who are well trained to this business. So there are disconnects in the system that still have to be addressed to get a better streamlined path forward.
Dr. Sangupta, do you have anything to say? Please unmute yourself. You are muted.
Yeah, I agree with what actually my connection got lost. So some of what Professor Chatterjee said, I couldn't hear, but yes, this exposure, the students, I agree with both of the speakers that there should be a strong background of any subject, and then the student should listen to the problems. I mean, it's not that I don't know the method or how it has been done. The question which is being addressed in any seminar, if they listen and if they get interested, so that I also find that students, they tend to think this way, that that this is not related to my work, or so that kind of mindset should not be there. So yeah.
And also what Professor Chatterjee mentioned, that apparently that difference in our education system, like when it's coming to math or physics, it's more of, problem solving, and the way in the school biology is being taught, that's more of, rote learning and just fact-based things. So I mean, this is something.
And I agree with Professor Chatterjee what he said, that the physics teachers in IIT Kanpur, they were not happy, as he said, and same as in chemistry, the physical chemistry teachers, I don't think they feel happy if the students go to biology, and it happens. So yes.
Okay. So before going to another question, I want to pull out on a question from chat box. It's very interesting. This is from Abhijit Majumdar from IIT Bombay. He's asking that we see now that all roads are leading to biology. What about the reverse? What about students trained in biology taking up engineering, physics, chemistry for that matter, these problems, and applying their training to other non-biosystems? What are your views?
Well, I mean, there's nothing stopping it from happening, right? It's again, I think that if you learn how to learn about a problem, tackle a problem intellectually, then you can work it out. The rest of it can be worked out, right? So the except that I have from my own personal experience, and people can correct me if I'm wrong, is that the later it gets, I've seen that the reverse becomes a little difficult, because there are some core mathematical skills and the ability to deal with certain concepts and skills in mathematics and physics and chemistry and engineering that are learned early, better learned early. And if you now have to go and acquire that kind of analytical skills, having been trained in traditional biology first, at least in India, it turns out to be quite challenging, although I've had students who have done that very, very well, right? So I'm not going to say it's impossible or difficult. It's definitely possible, but it's a little, the energy barrier, I think, is higher in the reverse direction than coming from physics or engineering to going to biology. but I've been proven wrong on that several times in my own lab. So there are exceptions to that.
Amazing. Dr. Gundia? You have already answered in chat.
I did answer a little, but
yeah.
I can add. So I had a student who came into my lab as an int biology major, and so he wanted to work in my lab, and I was, and initially I had hesitation, but I was proved wrong. It takes a different kind of student to do this kind of work. My student, he didn't know how to write code to start with, since he had a biology background, but he got trained, and now he works, after his PhD, he moved into industry, and he now works in a company that makes testing frames to test how tissues. And so he knows a lot of the, not only the mechanical engineering terms, he also knows a little bit of electrical engineering on what kind of motors will work in certain situations. So I think that the reverse is certainly possible, but it needs, for this kind of an interdisciplinary background to exist in labs. If I expect a lab which is doing purely biology kind of work, they will not be motivated to make that transition. So I think you're constantly challenged in environments such as ours, because the biology students are challenged to learn more engineering, and the engineering students are challenged to learn more biology. So I feel such places are the best where you can make this jump from biology to engineering and vice versa. I find it a little more of that energy barrier if you're working in a traditional lab and you suddenly decide one fine day, I will make the transition. So, but I do see it is possible.
Very insightful and encouraging. Dr. Sangupta?
Yeah, technology developed from biology, many technologies developed which was motivated by biological systems. Whether students can come, yes, what Dr. Namrata said, that they need to be trained. If they are motivated, they need to be trained. As Professor Chatterjee said, that there are some difficulties of mathematical understanding and all. So if a hardcore biology student, if they are motivated, they just simply cannot jump and try to solve something. They need to be trained and train themselves. They need to know that. So yes, many technologies developed from taking ideas from biology, right? So biology students also can move to contribute their biological knowledge. But yes, they need some required training for that.
Very encouraging. So in a nutshell, it seems like nothing is impossible. If you're interested, you can do it.
Yes.
Next, moving on to our next question.
I actually heard one Nobel laureate's talk who said that in undergrad he studied English. And probably that is true for many scientists in other countries. So they learned English major or something, and then they came to chemistry or physics and got Nobel Prize in solving some biological problems. So that is definitely possible, I think. Not even biology, they are from English major. So yes, I don't remember, sorry, I forgot the name, but yeah.
Very motivating, very motivating. Thank you for sharing. Next question, what we have is, how do you envision the interdisciplinary nature of biological research in years to come? Professor Chatterjee, would you like to start?
Well, I think it's going to go in increasingly unexpected and surprising directions. Part of it's going to be driven by necessity. Now we are tackling large-scale problems, not just in neuroscience, but climate change and things like that, where you have to have multiple disciplines and strategies on board together to tackle even slices of a problem like climate change, right? I mean, it can come from oceanography to chemistry to physics to biology and ecology and everything in between, right? So some of it will be driven by necessity. And when there's an existential pressure on something in terms of trying to solve a problem, people line up, people queue up, the best minds are attracted to it. And so it'll have a natural process of going into new directions that were driven by necessity. The other is not always a practical necessity, but curiosity. And that's also going to be a major driver. Neuroscience is driven by that. Now neuroscience is increasingly driven by a desire to treat diseases, right? And the inputs will come from, all I can say is it's very perilous to predict the course of these things, because the inputs and the surprising breakthroughs will come from areas you least expect, right? That's the only certain thing about it, is that the inputs will be uncertain and from unexpected quarters. And there are tons of examples of that. Like Yamanaka's core discovery in stem cell biology is now transforming neuroscience in terms of disease models, right? Who would have thought that that's going to happen, right? And there are artificial intelligence is getting inspired by neuroscience and is impinging on neuroscience now, back and forth, right? And 5-10 years ago, nobody would have expected these things. Right? So I think expect the unexpected is your mantra, and enable enough people to hear about these problems and learn about these problems so that they can all come in. The more, the merrier, and expected the unexpected. Those are my 2 takes on the future.
Thank you. Now, Dr. Gundia.
I think I'm going to talk only about the Indian context. I mean, of course, this area is doing extremely well in all over the world, and it is going to grow. I'm going to talk purely from the Indian perspective, that we have the problem of supply and demand. And I'm thinking about where our students are going to get jobs after they graduate, which will also dictate the kind of areas they choose, because end of the we also have to think about where all these trained personnel are going to land up. So I think in India, partly from the engineering education, we have seen not enough companies who do work in the areas which I'm familiar with, which I will stick with, which is tissue engineering, biomechanics, devices, which require inputs from people trained in electrical engineering, mechanical engineering, and material science. But this is not as much the case in the US. You have a very thriving industry with Medtronic, J&J, etc., who are all the places where students would land up for jobs. And so there is naturally this system where we are creating a need for personnel trained in such positions. In India, unfortunately, this industry hasn't caught up as much. Unless we see that this starts doing much better, and there is a need for devices in India. We don't make half the things we need because everything is being imported. So not just the engineering, which I mentioned, we also need a lot of trained personnel in biology who are conversant with methods in engineering, who can bridge this divide. So I think that from the jobs market, if we start seeing more players in the Indian industry, I do think this will be the way that our field grows much nicer in India. But where the field is going to go, I'm thinking tissue engineering, we've talked so much about, I mean, science fiction is full of ideas of how we can create replacement organs for this, that, and the other. We're not there yet. And to me, that is something which will drive the next decade of where things should go. Is to find the need and fill the need. So I'll stop there.
Interesting. Thank you, Dr. Gundia. So it seems like AI and coupled with the technological innovation, and that's the future it looks like. Yeah, Dr. Sangupta?
Yeah, I would say just your question, what you asked, yes, demand and supply is needed here also. Demand is there in the biology field to get supply from knowledge from different fields. But yes, in practical sense, if you say in Indian scenario, it's true that the students, whether they choose one field, of course, that depends how they survive in future. So that is there, but whether the demand of knowledge from different field is there or not, it's of course there. So.
Thank you, Dr. Sangupta. Thank you for answering all the questions. I think it's high time we take some interesting questions from our chat box and pull it live. This one is from Avijit Majumdar. This is to Sumantra Chatterjee. What he's asking, okay, sorry, I'm very sorry. What Avijit Majumdar is asking, it's a question to all, that what Professor Chatterjee mentioned about developing nose for the cool questions. Any tips how to develop or train the nose?
I think it's something that Namrata mentioned a little while ago. It's just hear enough, maximize your exposure to things, which is a lot easier in these days, right? There's so much access to information. You're actually overwhelmed with information. So listening and hearing things out and not get bogged down by what Professor Sangupta said is that, oh, this is outside my subject, it's not my area of interest or research, and I'll stay in my lane. As long as you're not stuck in that groove, then I think just being open to listening to other interesting findings. And the other thing I think is very high risk now that I notice. Namrata mentioned Cosmos, and I read the first two books of Carl Sagan I read were Broca's Brain and Dragons of Eden, and we had no TV at home, of course, and so those books actually had a massive impact. I learned stuff from them, and I still go back and look at them to see why the hell did I like this part. Right? So I think getting to read stuff from these luminaries early on to stimulate your brain is crucial. Right? And that's where you start finding things. I'll give you one concrete example. I had to wait after coming to NCBS for the lab to be constructed, and it was a long wait. There's no animal house, there's no lab. So I sat around in the old ISC campus canteen, and I read a book called The Emotional Brain by Joe Ledoux, who was a pioneer in our field, who established the field of amygdala function. And he had an anecdote about a patient who, and that story essentially stimulated a question that has driven my research for 20 years, right? I honed in on that one little anecdote from a psychiatrist more than 100 years ago, and I found that very interesting. And I said, why is this happening? And I went and asked Joe, where did you get this story? He said, there's nothing more. What I have is what I have. But from that, incrementally, I built up a question, and it's stayed in good stead. It's been fantastic. So I think you just have to keep going till you find something that, follow finding your bliss, basically. That's it.
Amazing. Yeah, Dr. Gundia.
So I think I agree that reading has, there is no other. So yes, there is access to information. My worry is that we are skimming the surface if we think that we understand a lot by attending, hearing a lot of the TED Talks or, just doing this. There is no other place than what you would learn from in textbooks and I see the need for science communication. Someone like me, I was very inspired reading. For me, it was J. Gordon, The Science of Why We Don't Fall Through the Floor, is the sort of title that he had. Richard Dawkins, and also David Coleman. These are some of the people who greatly influenced how I thought about the last two, especially on how I think about biology and communication. So I think one needs to be more open, not just to hearing discussions, but also in sinking your teeth into some good science writing. And that is the best way, I think, to get us thinking about cool questions. I think we don't know where inspiration comes from, so we just have to be probing more and more and checking each of these windows. The reason why I got into biology is very hard. When you ask me this question, where do I put my finger on what was that one eureka moment? And most of us, you would notice, didn't have that eureka moment. It is a gradual transition until we say, okay, now I'll do it. But that has to happen only through reading and exposure to books, not just in core science areas, but through good science communication. And I think now we need to be exploring more and more science writers who come out from India. So here we are starting to see some good places where science is being covered in the media. So that could be one place, but we need to transition it into good writing at some stage.
Thank you. It's really noteworthy that you mentioned you rather demystify that thing, that eureka moment. Rather, it's more of gradual transition and evolution. I mean, great. Yeah, Dr. Sangupta.
Yes, I agree with Professor Chatterjee. I mean, you have to be exposed to questions, and it's like that. You have to train your nose. You get the smells, I should say, fragrance of all the problems, and you would know that which question is motivating you, and you feel the urge to solve it. So it's kind of training your nose is needed, right, Professor Chatterjee?
Yes, absolutely.
Great. Another question we have from Shonak, who is a member of our team. He's asking that will revising the undergraduate curriculum, giving weightage to all core subjects, will actually help a student to find his own niche?
Well, it's a tricky question. I mean, you can upgrade curriculum and syllabus as much as you want. The tendency appears to be loaded up with more of the latest trendy topics and buzzwords. And you have a huge ever-growing laundry list of these topics under the guise of so-called biotechnology, whatever. And I'm not sure that's really helping the students think about a problem and picking a problem and getting the core skills sharpened. And the other aspect of it is, of course, hands-on. In biological science, you need hands-on training, which is demands resources and funds, which is not available in most of these undergraduate places. So it's a tough one to comment on, because I think what is more important is to see if students can at least learn how to learn and maybe have less on their plate, core fundamental subjects, core skills that, for example, Namrata outlined. There's a few good things that you learn well, and focus on those, and then learn from those to see how you learn from those. And then realize there's a lot more to be learned, and biology is changing all the time. So you cannot mug up on everything and learn everything. Have some core foundations, and then be ready to learn as the subject changes, right? What was fashionable 5 years ago is outdated now in biology, right? Physics doesn't change that rapidly anymore. Chemistry doesn't change that rapidly anymore. So if you're in a younger subject, you cannot keep up with the latest findings and load your syllabus with that. Rather, you need good teachers who can train you in the core fundamentals, ways of thinking about a science, and then let the rest fall in place later on. We need to be able to relax about this a little bit and get our priorities right. And I think right now, the core curriculum and school education or high school education or even college education is too loaded on putting everything on the list, and that doesn't help the student at all.
Yeah, interesting. So Dr. Gundia, you have something to comment on?
I do actually. So I agree that there is a lot of emphasis on content, on increasing how much subject is packed into one. And I'm guilty of the same. When I teach a course, I will try and pack every lecture with a lot of information in my area. I think rather than thinking about, yes, and, biology is expensive. That's something I realized when I moved into biology, that I need to pack in a lot of money into consumables, which is not easy for everyone to have access to. It's easier to think of doing models and theoretical work because that doesn't require the amount of resources one needs to allocate for biological sciences. That being said, I think we are missing some core courses such as critical thinking, which needs to be taught at undergraduate level. Critical thinking is something that will stay with you regardless of which discipline you choose. The ability to take an argument and to slice it up into different parts and understand which part makes sense and which part doesn't make sense is actually very important in especially your world, where information could be both right and wrong. And you don't know which one is which unless you do a lot of homework. So, or, how do you, come to a right conclusion? I don't mean the right conclusion, but how do you conclude that, something is the way it should be done? This is one place I find is very lacking in our undergraduate education, at least from the training I have received and from what I have seen how US and other systems work. We don't give, and I would say the same about the doctorate degree programs. As a student, one forgets that the degree you're getting is a degree in philosophy. And most students forget about where is the philosophy in what you have done. So whilst we zoom in very close on a small part of the subject, and you need to be a master of that turf, there is no question that you shouldn't be, but you have to have the ability to zoom out a little bit and to gain a perspective on why is it important what you have done. And that can only come through courses in critical thinking and through more detailed discussions that one can have with colleagues. So that part I find should be fixed in the Indian education system at some level. Or, by the time you become a student and come to a lab, this is where we try and facilitate arguments, because, arguments in a very positive sense is to be a little more constructive. And I make my students always try and critique at least 1 2 papers. And I tell them that it's not just about, that you say, oh, this thing is a terrible idea. I said a good critique is one where you can offer positive criticism on what would you have done differently. Do you have that capacity to do that? And I find us not thinking as much about these kind of questions. So I would certainly like to see some classes on critical thinking be a part of anybody's curriculum.
I'd like to add one other point quickly before Dr. Sangupta speaks, is that I've also noticed something that struck me very early on when I started my lab in Bangalore, is that there's this mistaking technology for science. Okay, and that's a part of the other side of the coin of what Namrata mentioned, is that it'll be cool science if we have all the best tools and technologies, and we cannot do good science because we don't have the resources with the technology. And so mistaking science for technology is a very common mistake that we make and that the students make. And I think they need to be decoupled and delineated in ways that clear up the mind early on in these students in terms of the critical thinking first, and then see what tools can be brought to your service to answer a question. And those distinctions are not made early in the education.
Yes, I fully agree with Professor Namrata, also what she said, that our undergraduate courses are designed or taught this way as if that's the full knowledge, that's the kind of end of journey. And so the students, they know everything of what is written in the books, but beyond that, if you need to think and need to know that there are many questions still we don't know, and the answer right now, what your textbook you get may change at some point. So that critical thinking is really necessary if you go for in research. You want to address new questions. Otherwise, your study will kind of biased with some pre-thought notion. So,
thank you, Dr. Sangupta, and all the speakers for your insightful remarks, and they give us so much of time to discuss what to do, what not to do, and encourage our audience and us for, with motivating stories and journeys. And it seems like nothing is impossible. Whatever you wish to do, you can in the end. So with this, I'd like to conclude this session. And once again, thanks a lot for all the speakers for giving out so much time. And again, thanks. So with this,
thank you.
Thank you. Thank you very much.
Thank you. I got the chance to meet at least virtually to Professor Chatterjee and Dr. Namrata. So
Yes, indeed, it was good to see you all.
And I think I love the idea of this forum, and I hope you can do more of these, and we'll be happy to help.
Very nice.
It's really encouraging. Thank you again.
Thank you very
all.
much. Bye.
Thank you. Bye-bye.
Thank you. And before we sign off, so I want to mention that some exciting events are lined up. So next we'll be starting from 2 p.m. Eureka. Although it's Dr. Namrata already demystified that Eureka, but still human mind is very much stuck to Eureka moments. So that's why we have one session Eureka where participants have already joined from undergraduates and above, and they performed, and there are judges who will be selecting the best performance, how they have expressed their science in a very creative way. So stay tuned. So before we sign off, just a brief overview of our today's session, what we have planned. One important point to mention that in a Eureka session, there is a scope of live voting. So stay tuned and choose your favorite expression of science. So with this, we'll conclude this session, and we'll be right back at sharp 2 p.m.